here, and today I will be telling you all about my research on the effects of drugs on COVID protease activity. So, as a basic overview of what my project is actually part of a grant proposal, which means that it is a smaller part of a larger project. The larger project is more focused on finding computational ways to predict how enzymes and proteins will work, but my project is more specifically focused on how we can find inhibitory effects of specific drugs on COVID's MPRO enzyme. And so you might be wondering what MPRO even is. Well, MPRO stands for main protease, which means that protease enzymes can basically cut or snip what are known as substrates. And in the case of MPRO, that substrate is known as an NSP, which stands for non-structural protein. It's basically just this one long line of proteins known as a polyprotein, and because it doesn't have any sort of structure to it, it can't really do anything. So what the MPRO does is, when COVID attaches to a host human cell, it releases three things. It's RNA, which is genetic information, the MPRO enzymes, and its NSPs. And the UNPRO comes and it snips the polyprotein. And when this polyprotein is snipped, it can then fold and kink in specific ways to where it has structure, and then can go take that RNA to the nucleus of the host cell and start the creation of new virions or spread the infection of COVID. So what has been hypothesized by many researchers, not just us, is that if you can take a drug that can fit perfectly into the active site of the MPRO enzyme, then you can theoretically stop the MPRO from ever latching on to the NSP and therefore stopping the spread of COVID within a human body. And so you might have seen the word thrown around. I don't know if you've been into like COVID research or anything like that. There's something being thrown around called the Pfizer pill, which is basically what we're doing, where if you find a drug that can fit into that active site, then you can stop the spread of COVID because you're stopping MPRO. And so my research, I'm only studying one drug, but my partner research, Shwetha Sreenivasan, she's doing multiple drugs. And the way we're doing it, we have a couple different controls, but we're basically using light emission from genetically modified cells to see how much MPRO is being inhibited. And in order to make sure that not all proteases in the cells that we're testing are being stopped, because there are some good proteases, we're also testing on another protease called TEV, tobacco etch virus, which is a different protease that we want to make sure which can indicate if other proteases will also be stopped. And so research is obviously going to be important for the medical field. Like, if you're able to get patients out of the hospital faster, even if they're vaccinated, if you can give them a pill that will help stop the symptoms a lot quicker, then you can get people out of the hospital faster. And with pharma companies, even if our results aren't entirely conclusive that, yeah, this drug is going to completely stop the spread of COVID, pharma companies can go and see our results and decide which drugs they should try and chemically engineer so that they can fit into the active site of the MPRO better. So that's all I have for you. Thank you for listening.